Okay, everybody, so this is our electromyography lab for clinical biomechanics. What I'm going to do here is I'm just going to run through a couple of sort of demo things that we would do in the lab. Unfortunately, you can't be in the lab to do them. So what Ian and I did was we collected some data, and what we're going to do is provide basically like a quick little demo of all these different questions that Nick has outlined. And basically this lab is going to work sort of like a discovery lab where you guys are advanced enough, you have enough of a background knowledge on EMG and how to use lab chart that I'm going to do a quick little demo, but then after that, it's on you to sort of figure out how to answer these questions. And then obviously your TAs will help you work through those, but I'm just here to sort of supply the general content and not answer too many questions in this video. So if it seems vague, that's on purpose. Okay, so what I'm going to do here is I'm going to get going. So electrode site and placement and prep of the skin generally is sort of like the same every single time. But the reason that it's also the same every single time is when we're doing research or in hospitals or something, we generally follow these CENIUM guidelines. Don't ask me what it stands for, but it is a consortium of scientists in Europe that in the early 2000s sought out the ideal sort of placement sites, electrode size, electrode distance, skin prep techniques, and solved a bunch of these key issues surrounding EMG consistency. Because again, EMG has to be consistent if it's to be replicated over and over. And a lot of this obviously involves collecting it all within one session, but also if you're going to be testing multiple sessions, even though you're collecting a different MVC, maximal voluntary contraction each time for normalization purposes, you have to prep the skin and do everything the same. So all those come from this CENIUM site. If you just Google CENIUM, all those are there. And I actually just changed specifically the electrodes we buy to conform with these standards even more. So you have me sitting here, and I'll walk through just generally. So I'm going to do biceps placement first. And I would say the thing that no sort of uncomfortable undergrad does is really palpate the site, like use your intro to injury and your anatomy knowledge, but not just sort of look at the muscle and assume you know where it is. Somebody might have a torn long head of their biceps, so you only can do EMG on the short head of their biceps, for example. You only know that by getting them to contract, knowing where the biceps tendon is, knowing where it inserts, and then I give myself a rough estimation of where those electrodes are going to go, just so I know where to shave. When I use the alcohol swabs, those will come off, but regardless of if the person seems hairy or not, I generally don't have really any hair on my biceps. You have these sort of like minute little blonde hairs that will allow the electrodes to not stick as well, especially if it's like a scenario where you're having the person, like I've done a study where I had a person do max squats, and when the person starts sweating, the electrodes do tend to come off because unless you buy really, really high-end ones, they don't breathe very well. So once you shave the area, which also helps with abrading, you don't have to use like sandpaper if you shave as well, because it gets rid of some of that dead skin. I then prep the area with an alcohol swab just to get rid of any of the oils that may be on the skin. What that alcohol swab also does is removes a little bit of those pen marks that you put on the person. So keep that in mind if you're really unsure as to where you're going to be placing this. Let's say I'm doing glute medius and I haven't done that in a little while. I would probably want to mark once, then prep with the alcohol swabs, let it dry, then mark again. After that, what I'm going to do is prep the area with new prep. It's kind of like an abrasive gel. It almost reminds you of like the grittiest toothpaste you've ever felt. And you can see that I'm prepping there, rubbing it really well until the skin is sort of like kind of red. You can see that. And then what that does is that, again, just removes any dead skin cells, any little bits of hair that were left by the razor. And then what I do is I try and get all that off just with an alcohol swab again. We have these little tiny ones. Sometimes I like using the bigger ones, but generally you can see the alcohol swab. If it's coming away very dirty, it means it's still taking oil and dead skin off your skin. So then you can look at the alcohol swab to see if you've done a good job. If it comes away clean, you're probably pretty good. But I've done this enough times that I'm not going to remark my own bicep here. I generally know where I want the electrodes to be. But again, if you're uncomfortable, you can always remark sort of like around the edges with a pen. You can see I'm feeling it just to see if there's any moisture left on the skin or if it's too damp or anything. And what I'm doing is I got my electrodes ready. I didn't think, like when I touched it, that enough of the sort of grittiness of the new prep had come off. So I give it one more clean with the alcohol swab. And sometimes, yeah, you can use Kin wipes or any like non-linting wipe to get the remaining moisture off. So we're gonna fan that a little bit. And again, if you have a participant in there, you can just give them a break, talk to them for a second. We're trying to do this quickly on video. But then I'll take my electrodes one by one. And again, I do this almost always flexing generally in the position where the participant will be contracting. Because again, you want to like mark the area and have them in a correct placement. I put the electrodes on side by side. These electrodes are 3 centimeter electrodes. So you have an collection distance of 3 centimeters, 1.5 and 1.5 to the centers. The Cenium standards actually, again, call for a 2 centimeter inter-electrode distance. That's ideal. On a larger muscle like a biceps, that's not necessarily going to matter, but on smaller muscles, let's say I wanted to EMG Anconius or a forearm muscle or something like that, 2 centimeters is more ideal. So we've actually bought new electrodes. They just haven't come in yet. So that's essentially how you would prep the skin site for electrodes, and then you can place your electrode leads on. They're just little snaps that will snap onto those. So the first experiment, the little exercise that we're going to do here is the muscle active? I will just get out my pointer here, and we will, come on, can't have the pointer to play it. Okay, so we are going to play that. I'm just going to adjust the audio there a little bit quieter, just in case. Okay. There we go. Okay, so we're going to play that. And basically here you see Ian with EMG on his anterior and posterior delts. You can see him as he creates a front arm raise and then an extension in each plane. So now he's doing an abduction, a little bit higher, and then adduction is kind of hard. He's just letting gravity bring his arm down. And then what we're going to do here, so those were just all body weight. We're going to do a resisted flexion. There you go. We have a resisted flexion. We're going to do resisted extension. All right, and now we're going to have resisted abduction and resisted adduction. Okay, and before I let this video go too far, we realized that we didn't label this data after the fact, but I do want to say that, so here you have our, I'll get out with the pointer again. We have our channels of raw EMG, and then we have our RMS calculations up here. So RMS being that's more smoothed EMG, and then here we have our raw, very stochastic, positive and negative. So in channel 1, this is channel 3, and then the corresponding RMS would be in channel 1. So in channel 3, we have posterior delts, and then in channel 4, we have anterior delts and the corresponding smooth data in channel 2 here. So when you get the data supplied to you, I believe it is relabeled, but we had not labeled it at the time of collection. Know that channel 3 is posterior, channel 4 is anterior, channel 1 is smoothed posterior, channel 2 is smoothed anterior. Okay, so we'll go back to the video. All right. So then what Ian's going to do is he's going to go in and comment out what each one of those were, so you guys know that what each one was when you analyze it. So when you go in to take a look at the data, you'll know from these comments what each exercise was, but really what you're determining is each muscle active at the time. And I will let you determine the criteria on how to do that. So we'll do a quick little lab chart tutorial at the end, but all Ian is doing there is commenting that out, so you know when he was doing these movements, so you don't have to exactly have this video side by side when you're analyzing. Okay, so exercise 2 is how active is one muscle in comparison to the other. So what we're going to do here is play this. And we did a little bit different exercise here, we have me set up with EMG on my wrist flexors and extensors, on my FCR and ECR. So I'm not actually flexing my fingers all that much. I'm trying to keep my fingers fairly neutral and flex simply from the carpals at the wrist. And this is a bit more zoomed in, so we'll show it zoomed out after a while. But you can see here on the right-hand side that we did a little bit better job of labeling the raw flexors, the FCR, the extensors, the ECR, and then we have grip force up here because I'll be doing a grip force task for a different part of the exercise. So then we have in channel 5 here, we have RMS of the flexors, and in channel 6, RMS of the extensors. So Ian is doing my resisted maximal flexion task and a maximal extension task. So again, you probably want to use some of this maximal data to determine how active one muscle is to the other. All right, so once we stop that, Ian will zoom out to make it look a little bit more like the other data. So again, so you can see our raw up here and our RMS down there. So now that we have our max trials, or our maximal voluntary contractions, we say MVC for short, what I'm going to do is I'm going to perform just a sort of wrist weight general flexion and extension task back and forth a few times, and then you can determine how much one muscle is active relative to the other during this task. And then I'll do the same thing, gripping a grip force transducer at 30% of my max. So I don't know if we show it here, see in a second, whether we show the actual max for the grip force. I believe we do. So Ian hands me the grip force transducer, which is plugged into channel 1 there. Okay, so yeah, here's going to be my max grip force. Squeeze that. Take a look and see what we got. Ian will then auto scale it. And then again, you'll be able to see this data and it will all be commented out. But now we have my max grip force, my max ECR isometric, my max FCR isometric, and now I'm going to maintain a 30% flexion force, or sorry, 30% grip force while I go through a flexion and extension task to my end range. Try and maintain that 30% as best I can. I deviate out of it sometimes, but then this will allow you to see how active one muscle is relative to the other while both have some baseline activity. Excuse me. Okay, so that was exercise 2. We can now move on to exercise 3, so determining when the muscle is on or off. So we're going to use the data from exercise 1 and get you guys to sort of think through it in a different way. Whoop. Come on. Okay, so again, when is the muscle on or off? You'll obviously need to determine this with some specific criteria rather than saying, look at it, it's on, or look at it, it's off. So again, Ian is going through these different motions here. And you're going to go through and use this data and sort of what you know through reading different papers or research or even lecture that Nick has talked about, how you would know that the muscle is on or off. You obviously will need some type of criteria, and I'll give a little hint here. You'll probably need some type of threshold to determine whether it's on or off or not. But that's the data you're going to use for exercise 3 there. So that's the same video you've already seen in exercise 1. You're going to use that data as well for exercise 3. How active are the muscles is our 4th and final exercise. All right, so, got a little ahead of myself there. Okay, so I wanted to pause it here because what we had here was Ian doing some wide grip, narrow grip, and different push-ups. And we left that data in so you can analyze it. and we didn't have necessarily video of it because something went awry there, but we have it all commented out for you. So we have a wide grip and a narrow grip push-up. We have EMG on Ian's pec and tricep short head. So on this channel here, channel 3, you can see we have EMG on pec major. On channel 4, we have tricep EMG on the short head, and then channel 1 is RMS of pec major, channel 2 is RMS of the tricep. So we had our trials here, and then we are doing our maxes afterwards. So not the best experimental setup, but we realized that we didn't include maxes, which is a little wink wink nudge nudge when analyzing this data, what you will need. So I do a max pec by doing a resisted push-up because that's sort of all we have. We don't have we couldn't film with the bar, so I figured that was the easiest way to do our max pec. Now I do want you to note here, you will see some crazy looking data with a massive spike. Just take note, this is pretty typical in experiments, what happened and whether the participant was actually contracting maximally when you got that spike. So you can see he kind of touched the lead there. That's pretty common to get these artifacts. How the baseline shifts there will generally tell you that that's like a cable sway artifact, but obviously you're going to have to work around that to determine which part of the actual contraction you are going to use. So again, Ian is going to comment that out and normalize that a little bit, and then we've got the video of him doing the actual reps at the end here. So there's the narrow grip. He's going to pause that, comment it out, and then starting again, wide grips with some more external rotation. Okay, so the last and final exercise is exercise 5 is the muscle fatigued. So using some of the prior knowledge you have about the indicators of muscle fatigue in EMG, there's a couple different indicators, some that are spectral based, some that are signal amplitude based. And so what you're going to do is go through here and analyze the data to determine, with obviously some set criteria, is this muscle fatigued or not, and how fatigued. So in order to do that, we're going to use the grip task that I was doing earlier. So you can see here, I head up to that 50% contraction I was maintaining before. So we have the previous MVC of the flexors and extensors in that, I believe, exercise 3 data file. And now this time I'm just maintaining that 50% contraction for as long as I possibly can. Again, just to reorient you, we have grip force up here. I'm trying to maintain that plus or minus 5% target around 50%. We have my FCR EMG here, my ECR EMG on channel 4 here. Then we have our RMS of my FCR and my RMS of my ECR. So right about here, definitely RP is going up. You can see me start to sort of lean back and really bear down on it. This obviously isn't the most exciting thing to watch. Fatigue protocols, especially if they're not maximal, can last a little while. But that's that. And I think it was, what was it? Just over a minute, maybe just under a minute if you take away that couple seconds at the start. So pretty close to a minute, but what you can do is analyze that EMG data and determine, is the muscle fatigued? Okay, so for the second part of this video here, I'm just going to run through a brief sort of overview of LabChart again. LabChart Reader, so I'm using LabChart 8 here. LabChart Reader is going to look slightly different, but not all that different. But I just wanted to orient you guys to the program again and how you would go through and use it. But there is a lot of analysis videos online. And again, you guys are upper year students. You can definitely take a look at Google and figure this out just as quick once you have this little demo. So again, here down at the start, you can see I don't have a Power Lab plugged in. So that there would probably be green if I was actually collecting data. But what all we care about is that we have the data present. So here you can see that I'm using that experiment 2, where I have the grip force up on channel 1. I have raw flexor EMG on channel 2, extensor on channel 3, and then 4 and 5 are my RMS values of those respectively. You can actually see that generally EMG gets brought into channel 3, so channel 2 is hidden there. But for our purposes, we're just going to do that. You can always go in and check what your channel settings are by going Setup, Channel Settings, and seeing here what input you have into which channel. Again, the way these Power Labs are set up, inputs 3 and 4 are the default EMG ones, so we set other stuff up in channels 1 and 2 to take up that space, but we didn't want to separate the RMS flexors and extensors. So you can scale your data on an individual channel by either double-clicking the axes. So if you double-click the y-axis once, twice, 3 times, it will zoom in to different values, or you can use this little arrow at the top, drop that down, and click Auto Scale. So let's say I was zoomed in more than I wanted and had the baseline offset on this EMG value here. I could click the top and click Auto Scale, and that would bring that back into center for me. I could also zoom in by pressing these plus or minus keys. You can adjust the zoom on the X and Y, or sorry, on the X axis or the timeline by clicking this 1 to 100 marker. And if you want to zoom in, you go down to 1 to 50, you go up in these values. So then times 10 would be obviously very zoomed in. But if you want to get a more holistic picture of the whole experiment, you can sort of leave it at a 1 to 100 or something like that, depending on how long your experiment was. So in terms of what is going on with each channel, you can see the channel settings if you click on this little box here off to the right. So for our EMG, you can see that there's no calculation. But if you want to see what is going on with RMS, you can see that they have regular digital sort of filtering that you can apply. We do our RMS by just writing in the arithmetic equation. So taking the square root of the smooth data. So that's our root mean squared. So you take the square root of the mean and apply that to the signal. You can also apply other filters by going down into digital filtering and applying those if Nick talks about different high or low pass filters in the class, and then setting the cutoff frequency. So that's how you would apply those. You can always hit undo, Control Z if you do something wrong. And then if you're ever in scope view, that's for something where you're applying multiple sort of quick stimulations and you want a bunch of responses overlaid. We use this a lot in neurophysiological experiments or motor control experiments. You do the same stimulation 1,000 times and look for the responses and then average them. For a style of EMG collection like this, where it's like ongoing long-term EMG, you would never really use scope view. You're going to use this chart view up at the top. So now that we're back in chart view, it's like this longer side scrolling collection, not that overlaid collection where it makes every single one of these stop points, these blue lines overlay each other. We know that we can, if you want to write something in, you can always right click, add a comment, and comment something out that you would like. So you can write that in yourself. Again, Ian generally wrote in comments for the tasks that we did. So the final thing I think you'll need to know in order to analyze your data is how to highlight and select some of the data. So you can highlight data on the actual chart view here by scrolling over it and then clicking Add to Datapad. So that's one way. We'll check out the datapad in a second, but the other way is to use your actual cursor here. So you can see that your cursor leaves an X on every single channel, and then over on the right hand side here, see where it says no sampling. Watch what happens when I pull my cursor onto each one of the channels. So it will tell me the value at that time point for on that specific channel, up where it said no sampling, up to the top right, and I can't go over to it because the number will go away, but it'll tell you it for every single channel. So if you're looking for a value at a specific time point, So you say, oh, I'm looking for this baseline value right here at each channel, what that is. You can see on my right on grip force, it's negative 5.5%. And then up here, you want to see what my contraction was. It's 50% there. So it looks like we actually I set in the video 30% contraction. It looks more like we set it at 50 now that I look at the data. Okay, and then you would be able to do the same thing for EMG. The other way you can look at some data is if you want to see the difference. So what's the difference here in my grip force? I can grab that little M from the bottom left, the marker, and pop it up there. And then if I click on here, or just move it along like I was before, I can see the difference in the Y value between my M marker and my cursor. So that would be 51% difference there. Again, if I get rid of that and just pop it over back down, you can see that I'm at 51% there. But if I get that M marker back up, the difference is visible. Finally, at the very top here, you can see instantaneous either difference in time, so 13-second difference there, or you can see what the time was of that collection. So from that blue line, the time starts at 0, and then what is the difference in time from that collection? So 19 seconds from the start of collection. So I wanted to talk about data pad. If we go to our data pad view, we can see that some data has been added here. Let's say I want to delete that data. Ian has in here mean power frequency of the flexor. So you can add a metric here in terms of what you actually want to measure. So if you want to add any type of arithmetic or calculations, you can add a new channel and calculate that there. We have some set up, so it's the mean for any selection that you would have. So we have mean grip force, mean EMG flexor, mean EMG extensor, mean of the RMS, mean of the RMS extensor, and then we actually have mean power frequency already set up for you as well. Again, I'll let Nick explain what mean power frequency is in the class, but if you are interested in using that value for this, we've set that up as well. So in here you can see Ian went in, selected that he wanted a spectrum calculation, and from spectrum he selected mean power frequency. So the way we get data in here is to go back to our chart view. If let's say I want this trial right here, I could select on the x-axis, I would click and drag, highlighting it in sort of that gray tone there. I hit add to data pad, and then when we go look at our data pad, it has given us all these mean values for each one of those channels. And that's a quick intro into Power Lab, and that's how you're going to use it to sort of discover your way and maneuver your way through this lab. So yeah, that's the end of this video. Good luck with that.